So I'm getting some questions about whether there are slides. There are some very brief slides in B courses under the genomics. There's a CoV2 folder, but today's lecture, oh, you can't hear me.
I can.
We can.
Yeah, Tess, I think it's your speaker. In any event, today's lecture will really be primarily based on a lecture that was given by Britt Glounsinger. And what we're going to do is we'll watch it together. I'm going to take questions in chat. I'll explain this more once everyone's online. But we'll go through this together in real time, and you'll have access to that video. You actually already have access to it. I'll be right back. Dogs woke me up at 4 o'clock this morning, so I was getting some coffee. All right, it's 10 after. Good morning, everybody. This is, I feel out of practice already, having not been in touch with you for a few weeks. but I'm looking forward to today discussing the molecular virology of CoV-2. So this background slide that you can see is of the CoV-2 viroid, and this is a really beautiful painting by a scientific artist named David Goodsell, and I have his link down at the bottom of the page. Somehow my pointer doesn't seem to be visible, but hopefully you can see it, and I encourage you to go and look at his work. So before I get into the material itself, hold on, I'm trying to change something, just a second. There have been a number of you asking about the grade distribution in the class, so I thought I'd tell you what we know at this point. So this is a little tricky to calculate because the Big Quiz 3 exams are still being graded, but this is the best approximation that I could make based on the information we have right now. So hopefully you can see this distribution. On the, pardon me, on the x-axis, you see bins that are based on the percentage scores. So these are not total scores, these are the percentages based on the material that's been submitted already. I think I received one question asking whether the biggest bin would be given a particular grade, and we haven't made those decisions yet. So there's a standard curve that's applied to 104 historically. And historically, 104 from semester to semester tries to have about the same grade distribution. But I think that in light of the challenges, the unusual challenges of this year, we expect that the curve will be far more generous than it has been in the past. So I can't give you any specifics about what these particular scores will equal in terms of grades at this point. Pardon me. But the main thing is just to give you the opportunity to see where you sit on the curve at this point. Okay, one more thing I should say right now is that while I'm projecting these slides in Keynote, for some reason I can't access the chat function. So I hope GSIs will feel free to jump in and answer any chat questions that you might see. Okay, so today we're going to talk about the molecular virology of the causative agent of COVID-19. Now, this shouldn't come as a surprise. I mean, historically in this course, the last two lectures, or last three lectures, are on infectious diseases in light of the material that you've learned earlier in the course. Frequently, the types of diseases that are discussed are things like tuberculosis or HIV or other infectious diseases that often have a rich body of science behind them. But I think in light of the unusual circumstances in which we find ourselves, that I would focus on COVID-19 and the causative agent, which is SARS-CoV-2. Here, what I'm showing you is a brief schematic of the life cycle of the virus. And you can see in the upper left-hand corner, I wish that my pointer would work, but in the upper left-hand corner, you can see the viral particle attaching to a cell through a receptor protein called ACE2. The protein then, the virus then enters the cell in a vesicle. Its genome is released into the cell and is translated into a series of proteins. And then these proteins help facilitate the replication of the viral genome and the assembly of new viral coats, allowing the virus to create many copies of itself. You know, essentially it's tricking the host cell into making many copies of the virus, and then this is released. So today what we're primarily going to focus on is the left-hand side of this chart. Viral entry, the genome, the translation of the genome, the composition of the genome, and the replication of the genome. But we probably won't get much into the reassembly of the new viral particle on the right. So we'll save that for Friday. I am not an expert, I'm not a virologist, but fortunately we have a member of our faculty, Britt Glounsinger, who is. And so what I've decided to do is, rather than reinvent the wheel, we're primarily going to focus today on a really beautiful lecture she gave that has been viewed by hundreds of thousands of people because it's a really wonderful introduction to the molecular and cellular biology of this virus. Now, this is an academic lecture. It will be challenging for some of us. So what I'm going to do is try to field questions in the chat box as we watch this. We're only going to watch half of it. I will pause at certain points in the lecture to try to expound a little bit more on some of the points that she's making and try to answer more broadly questions that you might be raising. And then as I said, I'll stop about halfway through because I think that's about all the material we can take. And I suspect that we'll be close to the end of the hour at that point as well. I don't want to go past 10 o'clock. So if I somehow lose track of time, please interrupt me and let me know. We'll stop it there. Okay. So this is, for those of you who might be watching this later in the, recorded version, this is the link to the video itself. We'll be watching a slightly different version because she sent me the raw video and you'll see it has some interesting differences from what's posted online. And just to get you started, I wanted to give you a little bit of terminology that you'll need. If you remember, there was a syndrome called SARS, and that stands for Severe Acute Respiratory Syndrome. Because of the relationship of the current virus to the SARS virus, it's called SARS-CoV-2. The first one was SARS-CoV-1. This one that causes COVID-19 is called SARS-CoV-2, even though the syndrome it induces is different from SARS itself. Okay, so the virus is called SARS-CoV-2, and the disease, not the diseased, but the disease caused by this virus is called COVID-19. So we just want to separate those two terms. And then another term that you're going to hear is for the enzyme that is required for replication of the RNA-based genome. And this is an RNA-dependent RNA polymerase. So this will be referred to as RDRP. And then the last thing before we launch into the material is that I'd like you to do a few things before Friday. There's a 7-minute video produced by HHMI that I'd like you to watch on your own time. And I'd like you to look at these 2 articles. One is a Nature article on the race for a coronavirus vaccine. And the second is a focused treatment of a particular research group that has made an important advance in the development of a coronavirus vaccine. And I'd like you to have both of those ready for discussion on Friday. Okay, so now I'm gonna exit out of Keynote and we're gonna go to this video. Let me just look at questions really fast before we move forward. Okay, so you have hopefully found the slides now. But as I said, the major focus is going to be on this video. Okay, so now I'm going to start a new share. It's here. Abby sent me a message about how I can turn on my pointer, but now I won't be using Keynote, so hopefully this will work out. Okay, can you all see the first slide of this video? Hopefully. Okay, great. Okay, so remember this video is a talk that was given by Britt Glounsinger. It's as if you're having her as a guest lecturer. If you have questions as we go along, please post them to chat. GSIs, please feel free to help answer them if it's something you have a particular knowledge about. I will also try to field questions. And then, as I said, there are a few places in which I'll pause and try to provide a little bit more background. Okay, here we go.
presenting a lecture on the fundamental molecular virology of coronaviruses. These are viruses that have been circulating in the human population and in animals for a long, long time. We know of 7 human coronaviruses.
Okay, let me see if I'm seeing that people are having trouble with audio. Let me just see if I can fix that. Actually have done that before. I just need to remember how.
Hello, my name is Britt
Can you hear
Glonsinger.
it now?
I'm a virologist and a professor at the University of California, Berkeley, and an investigator of the Howard Hughes Medical Institute. And what I'm going to be doing is presenting a lecture on the fundamental molecular virology of coronaviruses. These are viruses that have been circulating in the human population and in animals for a long, long time. We know of 7 human coronaviruses. These are present in 2 of the 4 known genera of coronaviruses, the alpha coronaviruses and the beta coronaviruses. The 4 circulating strains of human coronavirus are shown boxed in red here. There are 2 that we've known about for a long time called 229E and OC43. These, together with the other 2, NL63 and HKU1, which were actually discovered more recently after the SARS epidemic, but are likely also thought to be circulating in the human population and not recently emerging. These 4 circulating viruses are the causes of, some of the causes of the common cold. Probably 10 to 15% of the common cold are caused by these viruses. We also know of 3 coronaviruses which have recently emerged into the human population through species jumping or zoonotic transfer. And these, of course, are the original SARS coronavirus, MERS coronavirus, and the newly emerged coronavirus 2, which is the cause of COVID-19. Each of these, like other alpha and beta coronaviruses, are thought to have a common ancestor in bat viruses. And this is different from the gamma and the delta coronavirus genera, which have common ancestors in birds. SARS coronavirus and MERS coronavirus, as I mentioned, probably came from bats, but it's thought that rather than directly jumping into humans from bats, they entered into the human population first through 1 or more intermediate hosts, animal hosts. For SARS, the intermediate animal host, or at least the main one, is thought to be civet cats. And for MERS coronavirus, the intermediate host is probably dromedary camels. In these intermediate hosts, the virus probably jumped from bats to these hosts, then underwent some rounds of replication in these hosts, and in doing so, acquired probably mutations that allowed the virus to then more easily transmit to the human population. Now, for transmission purposes, we don't know what the other intermediate hosts may be, and we particularly don't know what the intermediate host, if there is one, is for SARS coronavirus 2, the current pandemic strain. It's possible that it came directly from bats, but it's also possible that it went through 1 or more other animals before jumping into humans. I should mention that these are certainly not the only coronaviruses in bats. More than 500 coronaviruses have been identified in bats in China, and the estimates of unknown bat coronavirus diversity reach into the thousands, indicating that these are probably massively undersampled in the bat population. And I think this is also important because it suggests that this current pandemic strain of coronavirus 2 is unlikely the last we will see of coronaviruses. In particular, we have now had 3 zoonotic jumps of highly pathogenic coronaviruses into the human population in less than 20 years. And with the huge diversity of coronaviruses likely circulating in bats, I think many scientists and virologists are quite concerned that they will continue to jump into the future as well. And so very worthy of continual studying even after this current pandemic is finished. It's worth thinking about comparing the other two highly pathogenic zoonotic coronaviruses, SARS and MERS, to the current pandemic. So SARS, which emerged in late 2002 and caused a little over 8,000 cases and 774 deaths globally. This was an epidemic that lasted about a year. It was mostly brought under control in 2003, and the last cases were seen a laboratory outbreak related to 2004. But the epidemic is now ended. MERS, on the other hand, has caused a fewer number of cases, 2,521 to date, with 866 total deaths. This is actually the most pathogenic of the viruses, the one with the highest death rate of about 34%. Unlike SARS, MERS infections still periodically occur, and this is probably not through circulation in the human population. This virus does not transmit particularly well human to human, but these new infections are thought to occur from occasional recurrent spillovers from dromedary camels into the human population.
So I want to take a pause here and make a point that I want you to think about in the next few slides. Now, I think one of the challenges with CoV-2 is that it is unique, as all these viruses are. It has a unique genetic signature. It has unique features. However, it is related to other coronaviruses, and we have some knowledge of the dangers of coronaviruses, both based on those circulating coronaviruses that cause the cold, but also based on SARS and MERS. And so as we move forward, I think one of the incredible incredible themes that I get from this storyline of CoV-2 is just how rapidly the science is moving forward. And part of that is because we already know so much about coronaviruses based on what we learned from SARS and MERS. So as we move forward, you're going to see information about the spike protein and how various features of coronavirus transcription works. And I just want you to keep in mind that some of what we know is based on the fact that both specific research on SARS and MERS, but also basic research on coronaviruses in general, has helped to build a framework and context within which we can study CoV-2. Okay, is the crown of spikes a signature feature of the coronavirus is a question, and the answer to that is yes, and you'll see more about that in a moment.
So why is it then that the SARS epidemic was able to be brought under control within about a year, whereas we are clearly far from bringing the current coronavirus 2 pandemic under control? And there's several ideas that I've heard discussed on this, and I just want to bring up three here, comparing SARS to the current COVID-19 pandemic. First is that the spillover reservoir, as I mentioned, for SARS coronavirus was known. This is primarily the civet cat, and these animals could be culled then to attempt to break the chain so that there were not further transmissions from these animals into the human population. For COVID-2, as I mentioned, the spillover reservoir is not known. Second, for SARS-CoV-1, most of the human transmission occurred in a hospital setting. And indeed, these hospital settings were hubs of transmission for that epidemic. And so once this was recognized and the risk to the medical community was recognized, personnel were able to implement barrier nursing in order to stop transmission of that virus. Unlike CoV-2, which has not just spread in a hospital setting, but in fact there is widespread community transmission of this virus. And then finally, for SARS-CoV-1, individuals infected with this virus tended not to transmit until probably 24 to 36 hours after the onset of symptoms. And in general, there was a lack of asymptomatic cases as far as we know. And so this is really important from a contact tracing perspective and the ability to stall or inhibit spread of the virus within the population through effective contact tracing and other public health measures. Unfortunately for COVID-2, the situation is very different, both in that there are possible and likely maybe abundant either asymptomatic cases, and further screening will be needed in order to confirm that, certainly abundant mild cases, which are furthering transmission in the human population.
Now, keep in mind that this lecture was given about a month ago. So now I think that the evidence is pretty clear that there's asymptomatic transmission of CoV-2.
So for these reasons, and probably other reasons having to do with the molecular virology and epidemiology, the pandemic that we are experiencing now is very different from the one that we saw in 2003 with SARS. And as of this morning, is reaching nearly 400,000 total confirmed global cases. This is not likely reflective of the actual number of cases. These are just the confirmed cases through screening, but as we know, there are currently limitations in testing, so the actual number of presumed cases is thought to be much higher than this.
I think as of today, we're over 1 million. Is that correct? I think it's over 1 million now.
And as of this morning, so March 24th, 2020, we were reaching over 17,000 deaths, 17,252 deaths worldwide. So as you can see from this graph, unfortunately, most Western countries are on a very significant coronavirus trajectory, which is one of basically exponential growth. While some countries have been able to slow down growth and limit spread, this is largely not the case for UK, Europe, and the United States in particular, which shows a graph that is displaying clear exponential spread. And so I think we can anticipate that the number of cases are going to continue to exponentially grow in the future.
I tried to get an updated version of this graph. This data is no longer being plotted in the same way because it's become clear that really the trajectories are more relevant on a local context rather than at a national context. So this type of plot doesn't actually exist for current data, but I do recommend that you all go check out. This is from the Financial Times, and this guy, John Burns Murdoch, hopefully you can see in the bottom left-hand corner, has made a number of really useful plots of CoV-2 data that I think some of you will find interesting.
All right, we've heard a lot in the news and from experts about the epidemiology of this virus, about the spread of this virus, the transmission and the control. And so that is not going to be the focus of this lecture today. Instead, what I want to do is really drill down on the molecular virology of how it is that this virus enters and replicates within cells in order to amplify itself. And so I've broken down the lecture into 4 parts. The first part, I'm going to discuss how the virus is able to enter cells through interactions with the spike protein and host receptors. I'm then going to spend time talking about once it deposits its genome into cells, how does the virus replicate that genome and get its genes expressed? And there are some very unusual and interesting features of coronavirus biology in this section. I'm going to then move on to talking about some of the remarkable cell biological changes that occur in an infected cell, particularly involving membranes and the formation of what are called replication and transcription complexes during coronavirus replication. And then in the end, I'm going to spend the last few minutes talking about immune interactions that this virus has with, in particular, the innate immune system, as these are likely drivers of pathogenesis of these viruses in animal hosts and in humans. All right, starting with the structure of the viral particle and entry, we know that coronaviral particles are pleomorphic. That means they don't really have a defined structure. They've been looked at by cryo-electron tomography to confirm this. They also have what's called a helical nucleocapsid. So looking at the structure of the virus, I'm showing you here on the left, the nucleocapsid, which is shown in the center in brown, basically refers to the genome, which is a 30 kilobase, a huge for an RNA virus, huge genome, 30 kilobase genome of RNA that is of positive sense or plus sense. So this is RNA. When we say plus sense, that means it can be directly read by ribosomes in the cell. That genome is coated with a protein called a nucleocapsid protein that forms sort of this helical nucleocapsid. That nucleocapsid protected genome basically is encased in a lipid envelope that is derived from the host cell. Many viruses have a lipid envelope. In all cases, those lipids are taken from the host. No virus is able to make its own lipids, but many viruses make use of and steal host lipids for their replication and sometimes for their morphogenesis. And so that is the case for coronaviruses, where you can see there is a lipid envelope, which is studded with a number of viral proteins, the most prominent of which is the spike protein, shown in blue. This is the one that, of course, gives coronaviruses its name, for the corona-like either halo effect seen during a solar eclipse that looks like this, or a crown-like appearance of these viruses under the electron microscope. The spike protein, as we'll talk about in a minute, is critical for viral entry process. Additionally, in red is a membrane glycoprotein called the matrix protein. This is the most abundant protein on the outside of the viral particle, and its role is basically to connect the membrane to the nucleocapsid. So you can see in sort of the inset there that there is, this is a transmembrane protein, but it has a significant C-terminal domain which makes contacts with the nucleocapsid protein, and that's probably important for the morphogenesis phase of the viral life cycle when these virions are formed. And another minor envelope protein called E is present as well, also thought to be important for formation of these viral particles at the end of the viral life cycle. A little bit more about the spike protein. There have now been published a few different research papers showing structural information for the coronavirus 2 spike protein. And what this, I've pulled this from one of the papers, which is cited below, and what this is showing here is the structure, a cryo-electron microscopy structure of the coronavirus 2 spike protein overlaid, showing sequence conservation of related spike proteins from other coronaviruses that are basically plotted onto the SARS-CoV-2 spike structure. And these are then color-coded based on their level of conservation across these related viruses. And so what you'll notice from the spike, the spike is a trimeric protein. What you'll notice is that there are sort of two domains. There's this upper lobular domain, which is the receptor binding domain. This is the thing that engages the host cell receptor, and we'll talk about that on the next slide. And then in this domain, you'll see that there are many residues that are colored in sort of a teal color, and this indicates that they are highly variable. Indeed, the receptor binding domain in the spike protein is the most variable part of the coronavirus genome, and this tends to be common for viruses in general. This is a region of viruses that are under intense evolutionary pressure because of interactions with the immune system.
Okay, let's take a pause because there have been a number of questions. Now, again, I want to make the point that much of what we know about CoV-2 is based on the study of other coronaviruses. So, for instance, the spike proteins are widespread and they're highly conserved. And you can see on the right, this is a structure of a spike protein. And it's been color-coded in terms of how well conserved different residues are. So the purple residues are highly conserved. The teal ones are less well conserved. And those teal ones are in the part of the protein that actually is interacting with the ACE2 receptor. And so often in interactions, when you look at the molecular biology of the interaction between a virus and a eukaryotic host protein, they engage in what's called an arms race, in which there would be an advantage to the host if it evolves new versions of these host proteins that cannot be recognized by the virus. Obviously, it's an advantage to the virus if there are variants that can bind to these new versions of the host receptor. And so you find that those interfaces between viral proteins and host proteins are often evolving rapidly. Again, with the host, I don't want to say try because there's no sort of cognition in this process, but there's a selective advantage to evolving away from a sequence that can be recognized by the virus. And there's obviously a selective advantage to the virus if it can keep up with the evolving host protein. Okay, so there are some questions about pleomorphic The pleomorphic issue is about the whole viral particle, not just about the spike protein. So the size and shape of the coronavirus is variable. So if you see in the upper left-hand corner, it's approximately 125 nanometers, but there can be some variability there in terms of how many copies of the spike protein are made, how large the envelope is. So it's just not exactly the same from viral particle to viral particle. Does that help with the pleomorphic issue? There was a question about whether all coronaviruses bind to ACE2 receptors, and I'm afraid I don't know. I believe that it's relatively common, common enough that the ACE2 receptor was a good candidate for being the receptor for the CoV2 spike protein, but I'm not sure whether it's universal among coronaviruses. Are there any other questions before we move on? Okay, on that topic, when one virus enters a cell and stimulates the host to make more of the virus, are they all the same size or varying? I believe, again, not my area of specialty, but I believe that they can vary. Even the viral particles produced by a single cell. Is there a competitive inhibition between the spike protein and the original molecule binding to ACE2? Oh, what a great question. That's a great question. Does the spike protein change the structure permanently of the ACE2 receptor? I'm afraid I don't know the answer to the second question, but I suspect that there is a competition between the original ligand of the ACE2 receptor and the spike protein itself. Does anyone know more details about that, GSIs? Okay. And yes, this answer to the plus RNA is correct. It can be read directly by the ribosomes. Oh, well, not for transcription, for translation. Okay. Let's see, is the ribonucleoprotein complex in the diagram with the nucleocapsid with the RNA genome? Yes, it is. Any other questions? Okay, so we'll move forward. But these are, yeah, this is exactly the type of question and discussion I want to have.
The lower part of this spike protein is the part of the protein that encodes and possesses the fusion machinery that is important for the entry process. And you'll notice this in purple is much more conserved, and also that is sort of a classic finding that the fusion machinery tends to be very conserved. And tucked in the center of the fusion machinery is actually this hydrophobic fusion peptide, which is very important for being able to fuse the viral membrane with the host membrane so that the virus can deposit its nucleocapsid payload into the cytoplasm of cells. So what does this entry process look like? Well, as I mentioned, the spike protein is the protein responsible for engaging a cellular receptor. And this is, you can think of like a lock and key mechanism where the key is the viral glycoprotein and the lock is the cellular receptor. Different viruses will use different cellular receptors as a way of getting into cells. The receptor we know for both SARS-CoV-1 and for CoV-2 is the same protein. It's a cellular protein called angiotensin converting enzyme 2, or ACE2. And that binding to that protein is important, but it is not enough. You need a second feature to happen, and that is a proteolytic cleavage event. And this is carried out by a cellular protease called TMPRSS2, and perhaps others, but that one people have suggested is clearly involved for coronavirus 2 entry. So what happens is that the spike protein interacts with the receptor. This protease then comes and cleaves the spike protein. And actually, there are two cleavage events, at least two cleavage events that are known for SARS coronavirus and probably CoV-2. These cleavage events, the first one, what's happening is that the receptor binding domain of the spike protein is being separated from the fusion domain. And the second cleavage event, which is not shown here, is actually an activating fusion event that activates the fusogenic state of this protein. And so that allows then subsequent entry, which for coronaviruses may occur at directly the plasma membrane, may occur upon endocytosis, or may occur at both sites. That really hasn't been resolved. So the spike protein is really a classic class 1 fusion protein, and there are a number of viruses that have fusion proteins of this type. The best characterized are influenza, the hemagglutinin protein for that. There's Ebola virus fusion protein is also class 1. HIV fusion protein is also class 1. And so what I've outlined here on the bottom is the basic stages that are known to underlie the fusion mediated by these class 1 fusion proteins. So first, in the prefusion state, you can think of this as almost sort of like a metastable state for the fusion protein. And prior to proteolytic event that triggers the fusion process, this receptor binding subunit, which has not been cleaved off yet, basically you can think of as sort of clamping the fusion subunit and keeping it tucked away and inactive until the virus has encountered the appropriate host cell and it can be activated by these proteolytic cleavage events. So protease cleavage that we talked about then causes the receptor binding subunit to move out of the way, and that unclamps the fusion subunit so that it can then form a pre-hairpin that is embedded into the target membrane of the cell. And this occurs through the fusion peptide. The fusion peptide is a stretch of hydrophobic amino acids usually, which means that they can be inserted into the membrane. This pre-hairpin then starts to fold back, basically forming a 6-helix bundle and progressively pulling the cellular and viral membranes together to promote fusion. And the final post-fusion conformation in these class 1 fusion proteins is always a trimer of hairpins. And by this mechanism, then, once the fusion has occurred, the viral nucleocapsid with the genome payload can be deposited directly into the cytoplasm of the cell.
So I'm not sure how much you talked about membrane fusion earlier, but it's an energetically very challenging process for the cell. And so the virus really has to take advantage of this protein that helps fold back the membrane and create a vesicle into which the virus is actually encased. Someone is asking if I can explain more about the trimerized part. So let me go back. I'm not sure I can. Yeah, I'm not sure what I can say about it. It looks like the spike proteins are moved closer, but I'm really not sure about that. And then someone asked whether you'll need 2 spike proteins that are located close to each other. It is important that there are multiple spike proteins involved in this. It's also important often that a cell is actually interacting with multiple viral particles. Often small numbers of particles are not enough for infection. You have to have sufficient viral load. There's a question about what's the function of the cleavage again. So the cleavage actually reveals the fusion part of the protein. So if we go back even a little bit further, let's see. Okay, can you all see my pointer? I don't know if that shows. Oh, good. Okay, so this part of the protein in teal up here is the part that actually interacts with the ACE2 receptor. The protease then comes along and cleaves that off, revealing the fusion peptide. And this is the part that actually mediates membrane fusion. Does that make sense? So the spike protein, this part of the protein helps position the viral particle. That gets cleaved off and it reveals this fusion machinery that actually is the part of the protein that mediates the membrane fusion itself. Okay, so let's go back and
excuse me, now emerged from SARS-CoV-2 may occur at both sites.
Okay, let me give you a sneak preview here. So it turns out that Britt Glounsinger, when she was giving this lecture, probably had COVID-19. And so the version of this that I'm playing for you is different from the one that you'll hear online because they cut out her coughing fit, which you're about to hear.
It hasn't been resolved. So the spike protein is really a classic class 1 fusion protein, and there are a number of viruses that have fusion proteins of this type. So protease, it may also enable, excuse me, the genome payload can be deposited directly into the cytoplasm of the cell. Some early studies that have now emerged from SARS-CoV-2 indicate that there are some interesting features that are different between its spike protein and that of the original SARS-CoV-1. And the first difference is that scientists know from research with the spike protein of SARS-CoV-1 that there are basically 6 critical amino acids within the receptor binding domain that are necessary for interaction with the ACE2 receptor. And interestingly, 5 of those 6 residues are different for SARS-CoV-2 than for SARS-CoV-1. Nonetheless, CoV-2 is still able to quite efficiently interact with the ACE2 receptor. Excuse me. The second notable difference is that uniquely, SARS-CoV-2 seems to have acquired a polybasic cleavage site. Excuse me. And this polybasic cleavage site is interesting and important because it's predicted to enable cleavage by other cellular proteases beyond the one that we talked about. It may also enable efficient cleavage by the cellular protease, the TMPRSS2 protease, that is sort of the canonical one that's been thought about for this virus. And it's particularly important because insertion of the polybasic site in other viruses has been shown to increase transmissibility, particularly for pathogenic influenza viruses. So it's going to be important to figure out whether the same is true for SARS-CoV-2. Okay, excuse me.
Okay, so we have about 8 minutes left. We have not gotten as far. I thought we would finish this replication and gene expression section. We haven't started yet, and it's really cool. So I'm inclined to stop here. I'd love some feedback from all of you about whether you find this interesting and useful in terms of, should we keep going next? Okay, good. Okay, so what I would suggest then is, could you, I would encourage you to watch this part of the video, this replication and gene expression, before Friday. Do it, take one pass through yourselves, and then we'll watch it together. And so that you can ask questions in real time. And I think it will give you, because this part 2 is pretty complicated, but it's so fascinating. And I think it will be really interesting for you as you start thinking more about this virus. So why don't you do that on your own time? I've asked you to also read those 2 very short articles, and there's another short video for you to watch. And I think that will just give you a richer context in which to be thinking about this. Are there any other questions now before we log off about the material or about the class in general? Would there be a way for I'll get in touch with her and see if I can get these slides from her. And I'll do my best on that, yeah. Could I rediscuss the importance of the polybasic cleavage? Let's, so essentially, I think we don't know what the importance of this is. This is, of the polybasic cleavage site. Remember that one of the key features of this virus is that there's a lot we can learn about it by comparing to other known coronaviruses. But there are some things that are different. And so where you see those differences, you think they might help you understand why this virus causes the particular type of disease that it does as compared with, for instance, SARS and MERS. So I'm not aware of the functional significance of this polybasic cleavage site. It's just known that it exists, and it's a unique feature that I think scientists, virologists are looking at more closely. Other questions? Okay, so you have a little bit of homework, hopefully not too much, in preparation for Friday, and then we'll keep going with this video. And I hope we'll have some time to discuss issues of COVID-2 more broadly. And I will also be setting up office hours for RRR week, and I'll send out the time for that relatively soon. So really glad to be back with you for a couple of lectures, and I really am wishing you all the best. I hope you're doing okay, and I will see you on Friday. Take care, everyone.